plan for today, we are going to discuss the topic of speciation. A couple of reminders and announcements. There is no class on Monday. This is a 3-day weekend. So, yeah. Exciting for all of us. And so those of you, of course, who have discussion sessions on Monday, you should have received instructions from your TA. If you haven't, you should be receiving them soon. So in terms of what to expect and what to do, and of course any office hours that anybody has on Mondays will be either canceled or rescheduled, and there should be information about that. And speaking of office hours, my office hours are normally on Thursday. As I just mentioned before, I had a conflict yesterday, so I am instead having them today at 4. Maybe I'll see some of you there. Another announcement. The TAs have set up a left-handed seat request for the exams for the quarter. If you would like to be in a left-handed seat because you're left-handed, there is a sign-up sheet through EEE. I think the links are there, as well as information posted on Piazza. So please indicate if you would like to have a left-handed seat. I think the sign-up sheet closes on Monday, I think, in order to give them time to work on the seating charts and so on. So please don't put that off if you want to have a left-handed seat. Now, when we left off last time, don't worry, we're not doing a clicker question. I'm just addressing this clicker question. As you may remember, last lecture, the very last 30 seconds, we did this clicker question, and sort of an astonishing number of students did not pick the right answer. I personally think it was because I was rushing you guys. I don't think you're really that confused about the topic. So anyway, instructed the TA who's in charge of scoring clicker scores to actually do this one as any response counts, because I felt like you guys were all, you're welcome. Because after all, I don't really care what you know when you're so rushed that you're distracted. I care what you know when you're like able to pay attention. But anyway, hopefully, now that we can all have lots of time to consider this, let's consider why D, in fact, is the right answer for this, okay? What is gene flow and what is genetic drift? Completely distinct evolutionary mechanisms, okay? So gene flow is associated, so this is associated with keeping populations the same, right? Because if you have gene flow between two different populations, they're more likely to be similar to each other. So gene flow between populations keeps populations more similar to each other than they would be otherwise, right? Genetic drift is something that takes place in a small population and is driven by small sample size and just sort of random changes. So to put this in sort of just text, here we go. So gene flow is movement of alleles, and of course the alleles are packaged inside individuals. So if individuals leave one population for another, so it's movement between populations, it makes them more similar to each other genetically. So that's why, if looking at this, you would not pick answers like E, because populations would not be different from each other because of gene flow between them. They would be different from each other because of lack of gene flow, but gene flow promotes similarity, not differences. And so the only possible circumstance, in fact, when populations would not be more similar to each other is if there was absolutely no variation in a particular allele, then it wouldn't make any difference that allele if individuals are moving back and forth. So either won't make any difference or it'll make them more similar to each other. Completely different from genetic drift, which is if you have a small population, you can have random changes in allele frequency just by chance. They both start with G. In fact, they both start with G, E, and E, but they're completely different evolutionary mechanisms. So make sure you understand the differences between these two, right? And so again, if a few individuals go off and found a new population, this is a founder effect. It is absolutely not gene flow, because those individuals are not joining another pre-existing population and mixing with them. They're going to a new area where there were no individuals at all. Okay, speciation. So what is speciation? It is the process by which one species splits into two or more species, or to pull the definition straight out of the glossary in your textbook, speciation is defined as the evolution of two or more distinct species from a single ancestral species. So this gives me an opportunity actually to emphasize the fact that there is in fact a glossary in your textbook, and if you hit a word you don't know when you're reading, then this is probably more useful, honestly, to go to the glossary to understand exactly how your textbook authors are using the word rather than use a dictionary or Google, because it might be used in a slightly different way. Certainly you can do that as well. But I do encourage you to use the glossary so you understand exactly how terms are being used in your textbook. So then in order to understand this actual process, then we're not going to understand the process until we first understand what is the definition of a species and how do we recognize one. And so the first thing we hit here is that there are multiple ways of defining a species. Of course, students are not very fond of this. You know, I think students often feel things like, well, why do we have 3 here? Why can't we just pick the best one and learn that? Why are we being bothered by distracting information or repetition in other kinds of ways of defining species? Well, the issue with defining species, as you'll see as we work through this, is that in fact, not any one of these concepts actually works in all circumstances. And so in practice, biologists find that they need to pull in a couple of different biological concepts. So instead of having one big, long concept that actually just involves all 3 of these, they're usually noted as distinct criteria for recognizing species, all of which have their own place in certain circumstances. So, so this is part of the discomfort that you may be having with a somewhat complicated situation, and all I could say is this is part of intellectual maturation, right, is to learn to deal explicitly with uncertainty and limits of knowledge and so on. That's a really important thing to learn in using these definitions and concepts, is to understand limitations. So, biological species concept, the morphospecies or morphological species concept, and the phylogenetic species concept, and we'll walk through all 3 of these. But let's also go ahead and visit the glossary again and see what the definition is of species in the glossary. And here it is in front of you right now. It is defined as an evolutionarily independent population or group of populations, and its species is generally distinct from other species in appearance, meaning the individuals are distinct in appearance, behavior, habitat, ecology, genetic characteristics, and so on. Which means, of course, we have to understand what a population is because it's part of the definition. So let's go ahead and pull that out of the glossary too. Here it is. A population is defined as a group of individuals of the same species living in the same geographic area at the same time. So, for example, a species that had a worldwide distribution might have a population in North America and another population in South America and another population in Australia, and or if for another species, there might be multiple populations even within North America. It depends on the natural history and the distribution of those individuals. So this will be more clear, I think, as we go through a couple of different examples. But let's go ahead and start with the biological species concept, which is the first one on the list, is also the most widely used. And the biological species concept identifies species based on a criterion specifically of reproductive isolation, meaning that if you have 2 populations and you're wondering, are these 2 populations, are they really 2 different species? How can we tell that if one asks, are these 2 groups reproductively isolated? Are they unable to breed with each other? Are they unable to produce viable and fertile offspring? If that's true, then they're considered to be different species. One of the reasons that this is also a widely used criterion is this also is the species concept that relates most closely to how speciation occurs in the first place, because speciation is thought to occur or be driven by reproductive isolation, which is the criterion for the biological species concept. So let's go through an example of this using a familiar taxon here, and that is dogs. We've talked about dog breeds before and how there are lots of different dog breeds. If you are familiar with dog breeds, then you know, maybe recognize some of these. This is just a handful of the ones that just happen to begin with the letter A. I pulled this off of the American Kennel Association website, which is the governing body responsible for keeping track of breeds and identifying them and so on. And there are about, there's somewhat over 150 breeds recognized by this particular organizational entity. And so are these considered different species? No, they're not, because in fact they can interbreed, right? In fact, these are called mutts or mongrels, when in fact these different kinds, so Afghans and Airedale terriers, can in fact breed with each other. The species, by the way, is Canis lupus, and this is the same species as the gray wolf, which is the species from which dogs are thought to have been domesticated quite a long time ago. Estimates range, so domestication of dogs is thought to have occurred between about 10,000 and 100,000 years before present. And some of these breeds actually are much more recent than that. That doesn't mean that 100,000 years ago people were trying to breed Afghan hounds, but rather domestication of dogs began at that time, and breeds have different ages. Many of the breeds are only hundreds of years old. But all dogs are considered the same species, and in fact they're considered the same species as the gray wolf. But dogs are considered a subspecies of Canis lupus, because it's still possible for dogs to cross or to breed with gray wolves, although that rarely happens in real life. So reproductive isolation, though, is in fact the litmus test, and so let's consider the different types of reproductive isolation. This is a figure that is not from your book, but I'm going to use this as a visual backdrop for discussing the same concepts which are gone through in your textbook, in this, but I think it's nice to have some concrete examples. So reproductive isolating barriers between two populations or two species are usually subdivided into prezygotic or postzygotic. The ones that are shown in front of you right now are examples of pre-zygotic barriers to reproduction, meaning that the zygote, these are barriers that exist before a zygote can even form, right? If you think of the zygote as when the sperm meets the egg, bam, we've got formation of the zygote, the single cell, usually a diploid cell, that is the result of reproduction. These are barriers that exist that prevent the sperm from getting to the egg in the first place, and there are many different kinds of such prezygotic barriers. So how to think of interpreting this particular diagram here, the blue is supposed to indicate one group and the orange is supposed to indicate the other group, and the question is, do these individuals come together, or is there some sort of barrier that keeps them reproductively isolated? Let's walk through all these different kinds. So one, for example, is habitat isolation. This is an example of a prezygotic barrier in which the organisms are reproductively isolated from each other because they never come in contact with each other. So this is an example from some snakes that are actually fairly closely related to each other, but they're considered different species because one is fully aquatic and one is fully terrestrial. And so they never come in contact with each other. They never have the opportunity to mate. Because they live in different habitats, this is described as habitat isolation. So these kinds of terms are actually at least fairly user-friendly. It is pretty obvious what this actually means. So this would be considered, again, prezygotic because mating doesn't even occur. Sperm never goes near any egg. Okay, what else? The next example here is temporal isolation, and this is the form of reproductive isolation that one gets between two groups when they breed at different times. Now the different times can be completely different seasons, different times of day or night, or even different years. Let's walk through this example here. These handsome creatures here are two different kinds of skunks. One is called the eastern spotted skunk, one is called the western spotted skunk, and just from their names you think, well, isn't this habitat isolation? Because after all, if one lives in the east and the west, and one lives in the west, they never come in contact with each other. In fact, their habitats do overlap. But the eastern spotted skunk mates in the late winter, whereas the western spotted skunk mates in the late summer. And therefore, they're not reproductively active at the same time, and so they never interbreed, and so they're completely reproductively isolated from each other, but the nature of the isolation is temporal, meaning time. So this is an example of different seasons. As mentioned, though, even times of day or night. So some insects mate earlier in the evening and some mate later in the evening, and this is enough also to temporally isolate different groups, different years. So a really fun example of this is from the extraordinary periodical cicadas, which have an unusual life history where for years and years and years they spend underground as immatures and just come up above ground every now and then. And at this point then they mate and then lay eggs, and then that's it for another several years. There are several different broods. Some are 13-year cicadas and some are 17-year cicadas because these are both prime numbers. This means that to figure out how frequently these different broods then are coming out at the same time, one has to multiply 13 by 17. Every 221 years then, if these periodical cicadas are in the same areas, they come out at the same time. You have massive amounts of periodical cicadas. At that point in time, there would be an opportunity for them to be around each other and actually reproductively interbreed, but the rest of the time they're out of sync. They're coming out a completely different year. So that's an extreme example of temporal isolation on the order of many, many years. So even if organisms are in the same place at the same time, there can be isolation by behavior. And so many animals in particular have rather elaborate courtship displays. This is a little video I'll show. I'll make it a little dimmer so you can see it a little better. These birds are called blue-footed boobies. They're kind of fun because they live in the Galapagos Islands, which are made famous by Darwin. And these individuals have this elaborate courtship display where they show off their blue feet. Actually, it's the male. So this male here has very attractive blue feet. And he's showing his prospective sweetheart over here on the left, this female, his attractive blue feet. They're also doing some wing movements and so on. But a big part of the display is showing off these blue feet. And so if in fact then this specific display then is a necessary part of mating for these blue-footed boobies, and so should a male not then show off his blue feet appropriately, then no mating is going to occur. So this then means that there is isolation between those individuals, those species that don't have blue feet or don't show that this particular form of behavior. So this is an example then of behavioral isolation. Hey, so what if behavior's fine, same place, same time? There can still be other forms of isolating mechanism that are still prezygotic, and one of them is called mechanical isolation, which just simply means that the parts are just not able to come together in actual mating. And so some famous examples of this are snails that are in mirror images of each other. So there are, in many species, all the snails actually are of the same type, meaning they're either left coils or right coils, because in fact snails with shells that spiral in opposite directions usually cannot mate with each other. And so in those circumstances, when one does have individuals with a different coiling direction, then this is reproductively isolating, and the isolation is mechanical in nature, meaning this is just a problem of geometry and getting parts together. So that's mechanical isolation. There can also be isolation at the level of the gametes. Many of the examples that are best known from this or have been most studied are examples in spawning marine invertebrates. So many marine invertebrates that live in the ocean then release eggs and sperm into the water column, and there's a great mixing, and the eggs have to be reached by the correct sperm of the same species. So obviously if this is all happening in the ocean, then there is no isolation unless there is some form of temporal or spatial in the water. But many of these species actually do have biochemical recognition at the level of the proteins that are on the outside of the gametes. So as a sperm reaches an egg, there is an actual recognition and species recognition that takes place biochemically. And so this is then what keeps sea urchin sperm then from fertilizing the wrong eggs. So this is again considered pre-zygotic because a zygote doesn't form because a sperm does not actually fertilize the egg. So now let's switch to barriers, reproductive isolating barriers that are post-zygotic. This is actually far less common, so there'll be fewer examples of this. If they're post-zygotic, then what that means is a zygote did in fact form. So just because a zygote forms, so it doesn't necessarily mean you get a healthy individual out of this. So reduced hybrid viability is one example of a post-zygotic reproductive barrier. So in this picture, for example, which is a salamander that is resulted from a cross between two salamander species, you can sometimes get actual individuals that are hybrids, such as the one shown in this picture. But if they're typically, if they are actually true species, then the hybrids are, if they are formed at all, they're sickly, they don't survive, they don't develop normally, for example. There are other, the other primary type of post-zygotic reproductive isolating barrier is just reduced fertility. And this is distinct from reduced hybrid viability because there are examples, there aren't very many, but there are examples where the hybrids are perfectly healthy seeming, but they're just sterile. And a very familiar example of this is mules. So mules, as perhaps some of you may know, result from crosses between donkeys and horses, which are considered different species. Mules can be perfectly healthy and but they are sterile, meaning you can't take two mules and cross them and get a baby mule. The only way to produce a mule is to take a donkey and a horse and cross the donkey with the horse. That's the only source for mules. So this is then a reproductive isolation between donkeys and horses because there's no intermingling genetically because a mule is a genetic dead end there. So those then are examples of post and pre-zygotic barriers. And so if you understood this, you should be able to answer this clicker question without any problem. We'll go ahead and just do this as any response counts. So consider two species of frogs. They both undergo courtship rituals in different locations. Males in one species make croaking noises to attract females on lily pads and ponds. In the other species, you have males in this other species that make identical croaking noises, but they're in fields, so they're far away from the ponds. What kind of reproductive isolation would this be considered? A, temporal prezygotic isolation, B, temporal postzygotic isolation, C, habitat prezygotic isolation, D, habitat postzygotic isolation, or E, behavioral prezygotic isolation. So if you're not sure, good time to chat with your neighbor. I'll give you 30 seconds or so and see if you can figure this one out. Let's try for 100% this one. Okay, looks like it has stabilized. Let's see here. Excellent. 86%. Yes. Okay, so looked like there was a handful. The one that was most common actually was number E. Why is that? Well, I think what got you students who chose E is you were thinking, well, this is a behavior, right? After all, they're croaking. Isn't that a behavior? Yes, that is a behavior. But actually, it's identical croaking, right? I mean, it's not the behavior that is isolating these individuals. It's pure habitat, right? So this is an example of habitat isolation, because the reason that these frogs, for the information that is given, the reason that the frogs are not crossbreeding is just they're simply in different areas, and so that's the issue there. So most of you got it. Good job. Okay, so why isn't this concept then just the only concept? Why can't we use it in all the time? Well, it doesn't always work. There are a couple of specific cases in which the biological species concept does not work, so let's go ahead and consider those. So for example, fossils. There's no direct way of telling if species that are only known from the fossil record would or would not be able to interbreed with individuals that are also represented from the fossil record. You can't observe their courtship rituals or any kind of mating behavior. So fossils then cannot be evaluated using reproductive isolation. Asexual organisms are also a problem for this because asexual organisms, and there are a variety of different animals, for example, that reproduce asexually. Some aphids reproduce asexually, little tiny insects that live on plants. And so if they're asexually reproducing, then they're already reproductively isolated because they're producing offspring without sexually encountering or interacting with other individuals of their species. And so if we just applied the biological species concept, then what do? Call each aphid its own species? Well, no, of course not. So that is why then the biological species concept is not very useful for asexual organisms. It doesn't work. Also, groups that don't overlap in time or space, it can be extremely difficult to evaluate their ability to reproduce or not. You know, how can you tell if an individual, if a population in North America can or cannot interbreed with a population from Australia? So sometimes it's extremely difficult to even evaluate that in the first place, which is why there are other biological species concepts. So the morphological species concept is a different species concept. So morphology, so this then involves characteristics that one can see and measure. This is the most common species concept then that is used for fossils. And so here's an example in front of you right now where we have fossils from two species in our own genus, which are considered distinct from each other because of morphological differences, meaning things that you can measure that are different between these two. Now, how can you tell? Is this not a subjective criteria? How similar or dissimilar do these fossils have to be before they're placed in the same species or not? You know, absolutely, there is a subjective element to this. And so a scientist that is making decisions about whether or not to name this as one species or two different species will lay out the criteria. I'm calling these different species because they differ in this particular way, and the scientist can draw on his or her knowledge of the kinds of morphological differences between other example species which are still alive and extant on the planet. So one can draw on other forms of information, but do you know for sure that this was the same species or not? No, that's a little bit gray. What one individual calls a couple of different species can be called the same species by somebody else. But again, the way that scientists communicate this is just simply by articulating the differences and why they do or do not find these differences compelling for calling these individuals different species or not. Phylogenetic species concept is being used more and more as more and more taxonomic groups even have a phylogenetic tree for them. I mean, one of the things that would stop one from using a phylogenetic species concept would be if you're studying a group and there is no phylogenetic relationship even worked out for your group, then you can't use a phylogenetic species concept because the phylogenetic species concept identifies species as being the tips on a phylogenetic tree, such as the one shown here. This is an example from your textbook which shows the relationship between a number of different groups of elephants. And you can see over here on the left the phylogenetic tree that best represents the relatedness between these different groups. So for example, African forest elephants are considered to be more closely related to the African savanna elephants than either one of these groups are to the Asian elephants. But at the level of these different populations, so for example, the elephants in Cameroon versus Eastern Africa, these are not so different from each other that they separate out as different branches on the phylogenetic tree. They're indistinguishable at the level of the characteristics that were used to make this tree in the first place. And usually one uses a combination of morphological and molecular characters for making a phylogenetic hypothesis or generating a tree like this in the first place. So these are coming out as groups, so these are certainly considered different populations because they are in distinct habitats, but they're not so distinct from each other that one can pull out huge differences. So these would be considered populations. So I'd like to alert you to table 27.2 in your book, which is a convenient way of reviewing then the pros and cons of these different species concepts. The 3 different species concepts are listed here along with reminding you of what the criteria are and when they work and when they don't work. So this, I think, is a very helpful way for you to review the differences between species concepts and why we have the inconvenience of having to have more than one in the first place. Okay, so that's how one identifies species in the first place. Let's now consider the actual process of speciation, the appearance of new species from an existing species. So we'll go through this sort of diagrammatically and conceptually. Okay, so speciation occurs then, it's thought to occur when populations of the same species, so we start with one species, but populations within this species become genetically isolated from each other. So if there's genetic isolation, what does this mean? Less gene flow or no gene flow. If there's perfect isolation, there is 0 gene flow. If there's just diminished isolation, then this would be less gene flow, right? Once they're genetically isolated from each other, then these isolated groups can start diverging from each other because of the evolutionary mechanisms that we've already been discussing, because of selection, genetic drift and mutation, and so on. So let's do this with a visual backdrop of a little cartoon here, which is in front of you right now, which is supposed to diagrammatically represent a big, one single species here. And we're just going to do this diagrammatically, thinking of a single gene. We've only got 2 alleles, very simple. But, oh, we suddenly have a barrier here. So we now have genetic isolation here. We have no gene flow. This population over here on the left is completely separated from this population over here on the right. And it probably makes complete intuitive sense to you that if we quickly get rid of the barrier, then things go back to how they were and they just mix again. But if they're isolated and they remain isolated, then what happens? Well, we have all these evolutionary mechanisms going on, right? Oop, looks like we just had a little genetic drift. Got some changes in the allele frequency. Mutation, right? How do I know this is a mutation? Well, we didn't see this one before, right? We have a new allele. The only source of new alleles is mutation, right? So there we go. Oh, it seems to be advantageous. We've got possibly, or maybe this is genetic drift. We'd have to stop and do an analysis here. But we certainly see that it is spreading in the population, which does suggest that this is advantageous. Ah, I got a mutation over here on the other population. And over time, they're becoming more and more different from each other. So hopefully it makes sense to you that given enough time, enough differences accumulate that then at a sufficiently later time, if the barrier then disappears for whatever reason, depending on what the barrier is, then if these two do come together again, if they're now sufficiently distinct from each other, then they will continue to be reproductively isolated even if they're in the same habitat. Maybe it's incompatibilities between gametes. Maybe it's behavioral isolation. Whatever it is, they can now, they would then be a different species if they can no longer interbreed. So populations then, how does this isolation come to being? Well, populations can become genetically isolated from each other if they end up occupying different areas, different geographical areas, or even if they start using different habitats within the same area. This is usually a very, very slow process, speciation. There is an exception, and this is if what's driving the speciation is a change in ploidy, a change in the numbers of chromosomes. And so this is something that happens in plants, but not so much in animals. In fact, it's extremely rare in animals, much more common in plants. And one of the reasons for this is because plants often have the capacity to self-fertilize, right? If there's an individual that is got a different ploidy number, if it's an animal, and then will only have offspring if it mates, then there's an incompatibility in the number of chromosome copies, then that individual is not going to be able to successfully mate and have viable offspring. In plants, if plants that have the capacity to self-fertilize, then different ploidy number, no problem, self-fertilize, and this gets around the problem of combining gametes that have different numbers of chromosomes. So that's one of the reasons that this is much more common in plants than in animals. And so then this can obviously lead to a very rapid reproductive isolation and a sudden change in polyploidy, but this is the exception. Usually speciation is considered to be extremely slow. Now, if you get nothing else out of this lecture whatsoever, I certainly hope that you are realizing that there is a continuum here in gene flow and how similar different groups from each other are. So this is not a figure from your book. I've drawn a double-headed arrow here to show the continuum between the two extremes. So let's consider the two extremes here. On the left, we have a case where we have two populations. There's extensive gene flow between populations. Individuals are constantly moving back and forth between these populations. They're virtually identical. If you took a sample, you wouldn't know what population it was from. Extensive gene flow, okay? This is one extreme. The other extreme on the continuum of speciation is shown over here on the far right, where you have absolutely no gene flow whatsoever between two groups. They are completely separate. And so these then would be considered a completely separate species. In between, you have cases where you have groups in which there might be some gene flow, and depending on whether it's a very small amount of gene flow or a more extensive amount of gene flow, you'll be on one end or another. And of course, the speciation occurs, with the exception of changes in ploidy number, then one can be somewhere on this continuum, and so where things go then depends on circumstances. The more gene flow one has between populations, the more slowly they will diverge from each other. And that should make a complete intuitive sense for all of you. Okay, so we considered that restricted gene flow leads to, restricted gene flow between populations leads to speciation. The more restricted it is, the more effectively or efficiently then this will lead to speciation. The more rapidly this will lead to speciation. But there are different names given to different circumstances of speciation. We're just going to consider these two main categories, which are described in your book. This figure is not from your textbook, but I thought it would be a better visual backdrop. The two different forms are allopatric and sympatric. These are different names given to circumstances where there is complete physical isolation between the two groups versus the groups being in the same geographic area. So let's start with allopatric speciation. This is considered more common. It's also a lot easier to understand. So if we consider an example of how allopatric speciation might occur, let's consider this example right here where we have a lake. And at this point, we have some species of fish shown here, a little sort of goldish colored fish, and they swim around in the lake, and we have basically one big population that's interbreeding freely all over the entire lake. Then there's a drought, and the water level drops, and as shown over here, because the bottom of this particular lake is not perfectly flat, then there are barriers that now sort of arise and stick up out of the water because they are now exposed. And of course, this sort of thing can happen in changes in water level, and individual lakes can turn into more than one lake. And so shown over here on the left, then, if this persists for a very long period of time, we've got reproductive isolation of the fish on the left from the fish on the right, assuming that over very long periods of time, you can get divergence between these two reproductively isolated populations, and because of genetic drift and or selection and or mutations. Eventually, we've got changes that occur, and you can see this, they're shown as different colors, for example, in this particular figure. If speciation has in fact occurred, then we will know this because when they're brought together again, looks like over here, some later point in time, over here on the right, water level has gone back. We're back to one big lake. Now these fish are mixing together. If they truly are different species, then they're going to remain reproductively isolated, and we now have 2 species in this lake, where before, much earlier, we only had 1. So this would be then identified as a speciation event in this particular case. So that's an example of allopatric speciation. The example of allopatric speciation in your textbook is a nice one, so I'm also going to spend a little time on that, and that is the example of snapping shrimp on either side of the isthmus. This particular figure is not from your book. I thought this might make it easier for you to know where we were than the figure that is in your book. So an isthmus is a word given for a land bridge, not this specific land bridge, but any land bridge in general is referred to as an isthmus. So the land bridge, which is Panama, formed approximately 3 million years ago, and when this formed, it connected North America and Central America here on the north side with South America over here on the southern edge. And so this generated a huge change, of course, for the terrestrial organisms which now could cross this land bridge. And so huge changes were seen at this time in the flora and fauna of this region as they were now able to cross this bridge. But just as the terrestrial groups are now suddenly joined up in a way where they weren't before, well, suddenly in a geological sense, right? It's not like, bam, there's Panama, right? Right? Suddenly over, many, many tens of thousands of years, right? But at the same time that these two are now connected, notice what's going on with the aquatic environment. Before the formation of this land bridge, we had free access across this same area between the Caribbean and the Pacific, right? And now all of a sudden, we've got a barrier. And so this barrier then affected all of the organisms on both sides, or on either side of the Panama land bridge. One of the groups that has been studied are the snapping shrimp. And so let's look at the phylogenetic tree, which is provided for your convenience in the textbook, and this is what it looks like. So here's a picture of one of these handsome creatures, and here is what the phylogenetic tree looks like for the snapping shrimp. In every case, for a snapping shrimp on the Pacific, there's a sister species on the Caribbean. And so there's a total of 6 different pairs of sister species. And so students find this confusing sometimes how to read it. So let me remind you that, of course, time is going like this from left to right. And so if we roll the clock back 3 million years, before they were separated then, that's just basically chopping off this part of the tree. Let's do that. There we go. This is what it looked like before the formation of the land bridge. We had 1, 2, 3, 4, 5, 6 group of shrimp, 6 species of shrimp that could go back and forth between the Caribbean and the Pacific without any issue whatsoever. And then once the land bridge formed, there was a split, right? For each one of those, because some were trapped on one side, some were trapped on the other side. These are reproductively isolated populations, and in fact, that is borne out by analysis of the species that now exist 3 million years later. These are distinct species, by the way, 3 million years later. Okay, so that's allopatric speciation. How about sympatric speciation? For a long time, allopatric speciation was considered necessary. It's like, well, you've got to have reproductive isolation to drive the speciation events, so we've got to have separation. So sympatric speciation, where you could actually have speciation in a single area, was considered highly controversial for many decades. It's now much more accepted to the point where it makes a regular appearance in textbooks such as your own. And also, there is a growing number of examples. Many of them are from plant-insect associations, because it's recognized that even if organisms are in the same area, they can occupy space differently. So, for example, if you have insects occupying completely different plant species in the same area, then they are separated from each other in the sense that they're hanging out on different plants. And one such example that is one of the best studied is the story of the hawthorn flies and the apple flies. So let's spend a little time talking about that. So this is an example of the hawthorn fly up here on the left. This handsome creature is an insect, and it is native to North America, where it has always lived on hawthorn trees, which are also natives. Then, in the 1600s, the apple tree, which is an exotic, was introduced to North America. And it was noticed that I'm not sure exactly when this was noticed, 100 or 200 years later, so the 1800s or 1900s, that there was a pest on the apple tree that were these flies, which were referred to as apple flies, that looked really very similar to the hawthorn flies, except there were some behavioral differences with the hawthorn flies. These are pests because the flies land on the fruit, whether it's hawthorn or apple. They land on the fruit and they lay eggs. These eggs hatch, and baby flies are maggots. And so if you have maggots then infesting your apples, this is not considered a good thing, and that's why they are considered pests, although we don't eat the fruits from hawthorn trees, and that's why we don't care if there are maggots in hawthorn fruits. So this has obviously then only been going on for a few hundred years, so it's not very much time. So scientists got very excited about this because they recognized that this could be an example of sympatric speciation sort of in action. So this has been very well studied. I'll just go through one example of some of the differences that are shown here. So in nature, you can have apple trees and hawthorn trees in the same general area, but you'll find the apple flies hanging out in the apple trees and the hawthorn flies hanging out in the hawthorn trees. So they are occupying different microhabitats within the same general area. And they behave differently. What does that mean? That means if you collect a bunch of apple flies and you collect a bunch of hawthorn flies and you bring them into your lab and you evaluate different characteristics, such as, given the choice between the smell of apple and the smell of hawthorn, where are you going to go? These are the kind of differences that are seen. So the x-axis here, or the bars shown here, are apple scent, hawthorn scent, both mixed together, or no smell whatsoever as a control. And these are the kinds of data that have been collected. So up on the top, we have the behavior of the apple flies. You can see that almost all of the individuals, the apple flies, fly to the apple scent, not the hawthorn scent. And similarly, or conversely, the hawthorn flies show the opposite behavior, where they respond positively to the hawthorn scent and not to the apple scent. So they are behaviorally then going to the different fruits, they're responding to different cues, they're separating out, and I will give you a clicker question on Wednesday about hawthorn flies and apple flies. Go off and have a fantastic 3-day weekend. I'll see you on Wednesday.